Hello, good morning. Today I will be doing experiment number 12. The title of this particular experiment is the titration, unknown acid-base titration. In this particular experiment, a student will be performing different activities in order to understand pH, the use of indicators, the necessity to measure pH, and why it is important to know exactly how how the reaction works and how to determine the strength of the unknown acid, as well as to use a pH meter and then plot a graph and find out the required objectives for this particular experiment. I must say that this particular experiment is a long one, but first and foremost, I would like all students to go back and read the introduction very, very carefully. This is a well-written introduction for this particular experiment. In that, it defines each and every item that is there, why and what is the rationale for doing this particular experiment, and then performing the experiment that is there. And it is possible that at the end of this experiment, you will find under the observation table a homework sheet which must be completed and attached to your lab report. So there will be no extra homework sheet from external sources in the experiment folder for E12. With that, I'm going to share my experiment with you so that just 1 second, so that I can find the experiment. Here, you can see it's the introduction to this particular unknown acid-base titration. You can see how well it is written. It says, what is the learning outcome? After completing this particular experiment, you should be able to use or measure the pH of various solutions, estimate the acidity of the solution using colored indicators, or basically indicators which will give different colors in acid and base, measure the progress of an acid-base titration with a pH sensing device or electrode, then graphically determine the endpoint and the new equivalence point. After which you have to calculate the concentration of the unknown acid. Now, there is a big difference between the terms endpoint and equivalence point. Equivalence points are always measured when we use our eye as the device to determine the completion of a reaction. When the eye is used to determine the completion of a experiment, then you must know that there is a limitation to which the eye can differentiate between colors. And therefore, there is always a slight excess of the acid or base added to ensure that the reaction is completed. On the other hand, at the end point, we generally assume that the reaction is completed at a very specific point. Such a point can only be determined by devices which are much more accurate than the eye. And so we can, after plotting a graph, determine the exact point where the neutralization occurred. And so with that, we can conduct the experiment. That is there. The first part, in the last one, basically before I go to the various parts that are there, is calculate the concentration of the unknown acid. Now, concentration can be expressed in terms of molarity, moles per liter, grams per liter, or in percentage. And they are all related to one another, where mole is defined as the mass of the acid taken over the molar mass of the acid, or it can be defined as molarity times volume in liters. So that conversion you will be able to use. This means that you have to write a balanced equation before you even start calculation, because in a balanced equation, you can write the neutralization reaction and it will give you the moles of acid required to the moles of base required in order to get a specific number of moles of water and the salt that is formed, because the reaction will be balanced. The pH and activity measurements, basically you determine the hydrogen ion concentration by looking, adding indicators, or by adding indicators and observing the change in colors. That is one way of doing it. Second way is to measure the pH, which is defined as the negative logarithm to the base 10 of the hydronium ion or the H plus ion. The letter P stands for negative logarithm to the base 10, and it is different from the natural logarithm. So we have to write log10. The pH range is between 0 and 14, and at the midpoint, the amount of H plus ions and OH ions in water is equal, and the square root of 14 is 7. So at 10 raised to minus 7 gram ions per liter, we will have the pH as a neutral pH, or what we call as the midway pH, at 7. Below 7 to 0 is acidic. Above 7 to 14 is basic. As the pH increases, the acidity decreases and the basicity increases. Using that, we can go through the whole setup for this introduction and then read it and then continue with the experiment. In my case, what I'm going to do as a demo is follow parts of each procedure and then show you how to do it. But I will not complete the experiment, but I will show you how and where to do the experiment and get your observations done. And so the first part is pH and activity measurements. So it says that you get 100 ml beaker, get a general purpose thermometer, a pH meter. Obtain a sample, unknown sample that you wish to determine the pH, and place about 50 ml of that in the balance. Lower the electrode into the solution and gently stirring, determine the pH of the solution. So there are two parts to it. First, get the equipment, and second, perform the experiment. So let's go and do the first part. So get a beaker, 100 ml, into that. We add a thermometer, add the chemicals, and we want to see which chemical we want to do. Okay, we can take whichever one we want. I'm just going to put it inside and say, note the volume, and then add the pH meter. And there you can see the pH is directly recorded at the bottom. At a specific temperature, and that is 19.9 degrees or almost 20 degrees Celsius, the pH of the solution is 30. Now remember, you have to copy this picture and paste it in your lab report. Then, so I finished the first part, pH and activity measurements. Now let's go to the second one. In this, it says set up two 50 ml tables. Label one as A and the label other one as B. So let's go and see. Get two test tubes. Oops. Get test tube. So you have one test tube and the second test tube. You put a label and write A on one. And remember, you can add up to three initials. So label one, and then you again label the second one as B. And whatever name and initial you want, it's up to you to add, but you have it. Then into this, you add 35 mLs of the stock solution. So go to the chemicals and then okay, let's go with the first one anyway. No, because it is A, we have to add A, and then we write the volume. Volume and say okay. And in the second flask, again go to chemicals. You add the base again. We will add 35 mLs and say okay. Now we will add indicator. The first indicator we are going to add, maybe we'll add. There are 4 indicators, and therefore you have to perform this experiment 4 different times. For each of the acids that are there, so we'll add 1, 2, 3, 4 drops of indicator, and then even in this you add okay, and in this also you add 4 drops of the indicator. And now you will see the difference recorded. Then in this way, you go ahead and continue with the remaining sets that they have asked you to add. I added 4 drops, you can add 5 drops, and repeat the other 4 unknown acids and bases, so you will get. Remember, for each indicator, in this case, the first indicator I added was phenolphthalein. Phenolphthalein is clear in the acid and has a pink or magenta color in the base. And so record each one of them. At the end of the experiment, make sure you copy everything, the pictures, and paste it in your lab report under acidity measurement with indicators. After that, we are going to do the titration part. And this titration part, you have to do it with 2 parts, because first part will be visual and the second part will be with an electrode or pH meter. So let's do the first one with the visual. And most of you should have done the titration in the lab. Erlenmeyer flask. They ask you to get 100 ml. Yes, 100 ml Erlenmeyer flask. pH meter. And then go to the chemicals, and they will say unknown acid. So unknown acid A, and I'm just going to add unknown acid A. They say that it's labeled as HC. 35 mLs of unknown acid A. So there you go. Then you add, go to the indicator, you add 2 drops of the indicator, you get the pH reader. We remove the pH, you get it, and then get a burette, 50 ml burette. Don't miss, please. Don't close the dialog box, titration dialog box. Click for chemicals, add 0.1 molar, so 50 mLs of the base inside. So remember the volume of base inside the burette is 50 mLs, but the starting point for the volume is 0 mLs and not 50, because 50 is at the bottom and not at the top. We have not inverted the burette. Now you go on titrating, adjust the speed at which you want to titrate, go on titrating, and keep an eye on the indicator Erlenmeyer flash, so that as soon as it changes the color, you have to close the stopcock. And you can see that the solution is still clear. As soon as it turns, it's going on halving. You see the color change. Once you see the color change, you write the final end volume, and the difference between the end volume and the start volume will give you the amount of acid, a base that was added to reach the end point. So this is one way of doing it. Okay, now I'm going to leave all this picture on the screen so that you really know what you're doing. So next, I'm going to again get, this time I'm going to get a beaker. I'm going to get a 250 ml beaker, because 250 ml beaker, I'm going to add 35 mLs, 35 mLs of the acid A, same acid I'm going to put, and now I'm going to include a pH meter on that, and then I'm going to get a burette, a 50 ml burette, and add chemical sodium hydroxide, 50 mLs, 50 mLs, and say, okay. Okay, now I have to click on this. Okay, I'm going to go to collect. Now you see, I have a pH meter there. Then I have filled up my burette. I have the 0 reading recorded over here below. So for 0 volume of NaOH, you have a pH of 0.96. Next, I'm going to say collector data. And then where did it go? Then I clicked on collect the titration data. Then I'm going to say view the titration data. So now I'm going to start the experiment. Titration dialog box. There is the titration dialog box already set up for the new experiment. I'm going to open my titration. And you will see that on the view box, you will see the line is moving as the ml is added. So the pH is being recorded. Now, if you want to record, I will show you how to find where this data is for you in order to plot a graph on the Excel sheet by yourself to do it. But this is how you do it. So now at the end of 5 mLs, you will see that a line is moving with the volume indicating. So remember, I'm not added any indicator in this solution. If you want, you can add the indicator, but without adding the indicator, how does one find the neutralization point? And here it is. Now, whenever you do a pH versus volume of titrant added, you will get an S-shaped curve. The shape of the S will indicate the nature of the acid that is being titrated. If it's slightly, it is exactly vertical between the two halves of the S, then we know it is a strong acid, strong base titration. On the other hand, if the titrations between the two halves is like a slanting line, then it shows that it is a weak acid. Furthermore, depending upon the nature of the acid, it is possible to determine whether it is a monobasic acid, that is, whether it has one hydrogen or two hydrogen or three hydrogen because of the number of peak levels that you will get or number of steps in the S form that is observed. And so you can say initially the acid pH increases very rapidly, but then you can see how it slowly increases. And so now you can see that the titration is carried out at 25 mL. Still, the pH is rising, but not rising to that large an extent. At the neutralization point, there will be a sudden sharp jump in the pH, and you can see that visually as the experiment proceeds on. So we'll have to wait till the pH is, the neutralization point is reached. One of the mistakes a student makes is that as soon as the pH is, the neutralization point is reached and you see a sharp curve, you stop the reaction. Don't stop. Let the burette drain out so that you get two halves of the S shape clearly indicated so that now you can see how quickly the pH is changing. And now you can also view the end of the pH titration. Now, at that point, it tells you that, the system is getting equilibrated, and you will see the two parts going almost simultaneously at the same time, same shape. And so you can literally see the alternate angles are same. And once at 50 mLs, you can stop the reaction. So keep an eye on the volume that is added. Then shut off the stop clock. Yeah. And so now you see, now you observe this curve. It increases very fast, then the pH changes very, very slowly, then starts rising up and then sharply rises up. And you can see that this is almost a vertical line in between. So midpoint, draw a line between the top part and the bottom straight portion, and the midpoint of that will give you the pH at the neutralization point. If you draw a vertical line below from the midpoint of the straight line, you get the volume required for the neutralization point. And so once you get the volume, you know the mLs of the titrant added, you know the volume of the molarity of the titrant that was used, and then you will know, you have already know the volume of the acid taken, and using M1V1 is equal to M2V2, you can calculate your exact molarity of the solution. Once you calculate the molarity of the solution, then if you multiply the molarity of the solution by the molar mass of the acid, then you get the grams per liter. And once you get the grams per liter, convert the volume liters into milliliters, and in order to get the percentage, multiply it by 100, and you will get the percentage of acid percentage of acid, which means grams of acid in 100 mLs of solution. And so this is your S-shaped curve. Now, if you want to view the data, then, so you got the graph. So if I were you, first I would copy this graph, because you can go under edit. It will say copy the graph and put it in your Word document. Then if you want to see the data, you just click on data and you will see exactly all the readings that were there. Now, if you want to copy this, paste it on your Excel sheet, then you can use it to plot your graph. That is there. And also by looking at this graph data, you can find out the exact neutralization point, because see, the exact neutralization point is 38.5, where the pH is 7. And after that, you will see that there is, so between 38.4 and 38.6, there is a sharp jump. And after that, you will see that as you add additional amount of base, the pH starts stabilizing because there is no more acid. And then finally, at the end of the experiment, the pH will be equal to the pH of what was the solution initially, of the base initially taken, not of the acid. And with that, I have shown you how to do the entire experiment. You can fill up your observation table. Let's go to the observation table before I stop it. You have the data, fill it up, generalized, and then volume reading, answer the questions, and these are your homework assignment questions. So there will be no extra additional questions, because this covers the entire experiment that you have done. With that, I'm going to stop the share and end the session.